Hi everyone, in this video I will be going through the structured questions for genetic expression and mutations RE. The first structured question gives you a real scenario. Cystic fibrosis is an actual genetic disorder where it is caused by the deletion of 3 nucleotides and it results in a non-functional protein. I hope by now that when you see changes in the nucleotide sequence, you can immediately relate it to why a protein function may be affected. Because DNA is first transcribed to form mRNA, and that mRNA is then translated to form a primary structure of a protein. This primary structure is so important, it can change a lot of things downstream. Primary structure affects the secondary structure, it affects the tertiary structure, and it also can affect the quaternary structure. All this affects the function of the protein because active sites may be changed, the original structure may be changed, the 3D conformation may be changed. So that's how we're going to frame our answer. When you look at the suggested answer and explain how 3 nucleotide deletion can result in a non-functional protein, the very first point that's mentioned is that when you lose 3 nucleotides, actually you lose one entire codon on the mRNA. And the loss of a codon results corresponding a loss of amino acids, one amino acids when you translate it to its primary structure. And when you lose an amino acid, actually you change the primary structure, which will affect the secondary, tertiary, and quaternary structures because the bonds that are formed will be very different. And when the bonds formed are different, what will result downstream is that the 3D structure of the protein will be changed. And when the 3D structure of the protein is changed, it can affect the function of the protein. Okay, so now let's look at 1b. State the type of variation exhibited by cystic fibrosis and explain your answer. This is something we've not quite gone through before, the idea of variation. There are 2 kinds of variation that exist. Okay, but maybe first and foremost, what is variation? Variation refers to differences in characteristics. For example, I can have skin color. Everyone has a different skin color, but notice that we can't quite separate skin color into, dark and light. In actual fact, humans race, every human being comes in a different skin color. There's a wide spectrum of colors. Another example perhaps is maybe height, right? There isn't a category where someone is short and then the other category someone is considered tall. There is a spectrum of heights. There's short, shorter, tall, taller, and so a wide range of different characteristics. Whereas there are certain characteristics where it is not so continuous. For example, when we look at blood types, you either get A, B, or O. There isn't a continuous range of differences. So actually there are 2 kinds of variation that exist. One, we refer to as discontinuous variation because this variation is very distinct. There are distinct categories. And then another category of variation is continuous variation. Continuous variation looks at variation that is continuous. There is a wide spectrum of different characteristics. And an example would be skin color, height, and weight. Yeah. So in this particular example, cystic fibrosis, what kind of variation is it? It's actually a form of discontinuous variation, where you either have an individual with cystic fibrosis or no cystic fibrosis. They are very distinct categories. It's either yes or no. Right? Also, cystic fibrosis is controlled by just one gene. and is not affected by the environment. Whereas if we look at continuous variation, traits that have continuous variation, for example, skin color, height, and weight, these are traits that are often affected by many different genes. Many genes can contribute to skin color, height, weight, and also these traits are also things affected by the environment. For example, a lot more sunlight can affect the skin color. How much you eat, your diet can affect the height, the maximum height you can go to, and also your weight. So these are continuous variation. Next, we look at question 2. Question 2 gives another real case study, sickle cell anemia. This is, and in sickle cell anemia is caused by a particular mutation. They've given you two sequences, one of hemoglobin A and another of hemoglobin S, which is the sickle cell anemia version of it. You notice that largely the amino acids are the same, except for if we compare the position of amino acid 6, you converted one amino acid to another different amino acid. And this difference results in a different kind of hemoglobin being produced. So 2A asks you to state and explain the type of mutation responsible for this change. You would either mention missense mutation because a different amino acid is produced, or if you mention base pair substitution is also fine. You would then go on to elaborate your answer. What exactly has changed? Well, you want to remember this particular case study. In your textbook, they've actually mentioned this before. Your textbook mentioned that what actually happens is this. After transcription, one of the codons on the mRNA is changed from GAA to GUA. And this results in a change from glutamic acid to valine after translation. Okay, this example is an example given in your textbook. And as a result, the sequence and the type of amino acid has been changed. Or in other words, the primary structure has been changed. And this will in turn change the polypeptide chain. Okay, so state and explain the type of mutation responsible. Here was the stating part. Explanation part are the later parts. Next, when we go to B, red blood cells with hemoglobin S have higher tendency to crystallize under low oxygen conditions, causing them to be sickle-shaped. And then this can cause fatigue and swelling of limbs. They ask you to suggest how this mutation may have resulted in fatigue and swelling of limbs. So remember, suggest questions are questions where you're open to suggest your own theory of how this may happen that is within reason and logic. In the case of why it may result in fatigue, well, if a red blood cell goes into a sickle shape, it would become inefficient in carrying oxygen. The red blood cells also are more fragile and hemolyze easier. Hemolyze means it lyse or break more easily. This one seems a little bit of a stretch. Maybe it's a little bit harder to derive, but the first one is possible. And if it's inefficient carrying oxygen, there will be insufficient oxygen supply to various parts of the body. So this may result in fatigue. Swelling of limbs. So this one is a bit tough to suggest, but one suggested answer is that sickle-shaped red blood cells may have a tendency to block capillaries more and obstruct the blood vessels, thereby interfering the circulation and may cause swelling in various parts of the body. When we look at normal red blood cells, actually they're quite flexible and they can squeeze their way through small blood capillaries. But sickle cell shaped red blood cells are not that flexible, a little bit more rigid, and so there's high chance they will all clump together and block blood capillaries. Look at C. With reference to table 2.1 and 2.3, state the genetic sequence of the template strand. Here they've given you just a segment of the genetic code with 6 different codons. They want you to refer to table 2.1 and then they want you to write out the genetic sequence. So if you combine the two pieces of information here, you'll be able to write down the genetic sequence. So we are working backward this time. From the amino acids, we get the genetic sequence. So to be able to reach the template strand, you must the first step backwards is to have the mRNA strand, right? This is the mRNA strand if we look at the genetic code. So we are kind of like reverse transcribing. Reverse translating. We see the amino acids and then we derive the codons. From there, you then write down the template strand, which is the direct complementary base. So C will have G, U will have A, G will have C. This will be the template strand, and the template strand runs from the 3' to the 5' direction. The coding strand, which is also the non-template strand, is also complementary to the template strand. So you will just write the direct complementary base. Notice that the non-template strand or the coding strand is essentially similar to the mRNA. The only difference is in the T and the U. Okay. Finally, in writing your answers out, right? This is one way you can write your answer. Another way you can write it is from here, this end, all the way to this end. In other words, the 5' end to the 3' end. Currently, what you've written in this box is 3' to 5'. If you write it in the 5' to 3', this will be the sequence. Okay, last but not least, this is a very interesting essay question. Contrast the process of transcription and translation. Once again, when writing essays that compare and contrast, you are allowed to write down and draw a table. Your table, however, needs to be very clear in what you're comparing. For example, here we put in several headers to show what exactly we're comparing. We are comparing the location where transcription and translation takes place. We're also comparing what the template is, what the direction of template being read is, the raw materials, so on and so forth. Let's quickly run through the various comparisons. First, transcription takes place in the nucleus, in eukaryotic cells, and translation takes place in the cytoplasm. Okay, or some may say ribosomes, right? But actually, ribosomes are found in the cytoplasm or bound to RERs. We made emphasis to eukaryotic cells because in prokaryotic cells, transcription translation takes place in the same place. Next, template. For transcription, the DNA itself is used as a template, or more specifically, the template strand. In translation, the mRNA is now used as the template, quote unquote, for synthesis of the primary structure. Next, direction of template being read. Actually, you read the DNA template strand in the 3' to 5' direction because the mRNA is synthesized in the 5 to 3 direction. And translation just takes that and immediately reads it in the 5 to 3 direction to synthesize the primary structure. Next, we look at raw materials. For transcription, we will need free RNA nucleotides to create mRNA. Whereas in translation, we will need free amino acids to create proteins. The end product of transcription is the mRNA, whereas the end product translation is a polypeptide chain. The bonds that are formed in transcription is the phosphodiester bond, catalyzed by RNA polymerase, and in translation, ribosome catalyzes the formation of peptide bonds between amino acids. Last two. In transcription, to be able to start transcription, we require a promoter to initiate the process. And to initiate the process of translation, we do not require a promoter. Instead, we use a start codon. Involvement of tRNA, it does not involve a tRNA for transcription, whereas in translation it does. So maybe one more thing you'd like to compare is how we stop transcription. To stop transcription, we would require a terminator, whereas to stop translation, we require a stop codon. Right? So these are the differences between transcription and translation. You may want to incorporate this table, a very succinct table, into your notes. And that's all for the going through of this particular RE on structured questions. If you have any more questions, please feel free to contact me.